Hello, I am Professor Sims, and this is the first of 10 lessons included in my Pathogenic micro course. If you are currently enrolled in this course, please consult the syllabus on the Moodle site for assignments and other course information. The learning objectives for this unit include describing physical and chemical defenses, the role of microbiota as the first line of defense for infection, how enzymes and bodily fluids provide protection against infection, the function of antimicrobial peptides, complement components, cytokines, and acute phase proteins, the classic alternative, and lectin complement pathways, hematopoiesis and functions of elements in peripheral blood, explaining the mechanisms by which leukocytes and phagocytes recognize, destroy, and degrade pathogens, signs and symptoms of inflammation and fever, and why those occur, and then we'll talk about the advantages and risks posed by inflammatory responses. The first defense tactic is to present barriers to would-be invaders. So this is going to be your non-specific, innate immunity. It provides the first line of defense against infection by non-specifically blocking entry of microbes and targeting them for destruction or removal from the body. Skin is one of the most important barriers. Microbes normally can't penetrate the skin unless there's a breach in the barrier, so this is a cut or scrape that exposes flesh and makes it available for colonization by pathogens. We can banish pathogens simply by ejecting them, so from your urine, tears, and saliva, these wash them away. In the lungs, we have little hair-like structures called cilia that sweep them up and out, and coughing and sneezing helps us get rid of pathogens in our airways. We also marshal microbes to fight microbes, so fight fire with fire. These have to do with our resident microbiota, our normal flora. We have trillions of microorganisms that normally live in and on us, and these are a formidable defense system for us and for other animals. They produce toxins and enzymes that they use to outcompete and attack invading microbes. There are multiple types of cell junction in human tissues. Figure 17.2 shows these. You've got these guys here, your tight junctions. These rivet two adjacent cells together, which prevents or limits material exchange through the spaces between them. Here you have desmosomes. Desmosomes have intermediate fibers that act like shoelaces that kind of tie two cells together. And some small materials can pass through, but larger ones can't. And finally, you have gap junctions. These have channels between two cells that permit communication and signaling. An interesting example of the tight junctions as a physical defense, this is going to be in your blood-brain barrier, and that uses a particularly dense suite of tight junctions to prevent microbes from entering the underlying tissue. Both normal and transient flora can be pathogenic if the skin layer of protection is breached. So if you have even normal flora that is on your skin, that it can get into your bloodstream and it can cause infection that way. This is Rose Gardner's disease. It's a rather gnarly case of Rose Gardner's disease. This occurs when the fungus named Sporothrix shinki breaches the skin through small cuts, and they call it Rose Gardner's disease because they discovered it from people that were growing roses and their skin was getting cut by thorns. And then the fungus got in there and caused this really nasty infection. Human skin has 3 layers. These are going to be your hypodermis, which is this one here, the dermis in the middle here, and then on the outside is the epidermis. The hypodermis is the layer of skin that contains blood and lymph vessels. The dermis is the one that has your sebaceous glands and hair follicles. Also, your blood vessels are going to be in the dermis and the hypodermis. The epidermis is the outer layer of skin. Microbes trapped in dead skin cells of the epidermis are removed from the body by mechanical means, such as shedding of dead skin cells, mucociliary sweeping, coughing, peristalsis, and of course flushing by bodily fluids. Peristalsis is a series of muscular contractions in the digestive tract. It moves sloughed mucus and other cells and other materials through the intestines and into the rectum, and then it's excreted from the anus via feces. Goblet cells produce and secrete mucus. The arrows in this micrograph here, this arrow here, It's pointing to the mucus-secreting goblet cells that have been magnified by 1,600 times their actual size. And this is in the intestinal epithelium. The flushing action of urine and tears are examples of mechanical physical defenses. These both serve to carry microbes away from the body. Obviously, your tears are going to flush microbes away from the eyes. Urine washes microbes out of the urinary tract as it passes through, and as a result, your urinary tract is normally sterile. The microbiome is a community of microorganisms such as bacteria, archaea, fungi, and even viruses that inhabit the gastrointestinal tract and influence metabolism and immune system. They also influence your appetite and even your mood. Your resident microbiota, these are the normal flora that occupy your cellular binding sites. They live with you permanently, and some of the advantages to having them is that they occupy cellular binding sites, which prevents pathogens from binding to your cells. They also compete for nutrients and prevent pathogen proliferation. It's important to know that your normal microbiota get depleted during antibiotic treatment, and this can increase your susceptibility to pathogenic invasion. Your normal microbiota contribute to chemical defenses, so you have some of your normal flora can produce their own antibiotics, which help to eliminate pathogens. Some chemical mediators are shown here in this table. Chemical mediators exhibit nonspecific defense. Well, the ones that do exhibit nonspecific defense can be produced either endogenously, which means that they're coming from within an organism, tissue, or cell, or they can be produced exogenously, which means that they came from viral elements, DNA sequences that came from viruses, and were ancestrally inserted into the genome of germ cells. So this means that part of your DNA came from viruses, millennia ago. Sebum is like an oily substance, so it's in sweat. So sebum comes from a sebaceous gland. The sebaceous gland is another name for a sweat gland. Okay, it's an endogenous mediator. It provides additional layer of defense by helping seal off the pore of the hair follicle, and this prevents bacteria on the skin surface from getting into the hair follicle or getting into the sweat glands and the surrounding tissue in the dermis of your skin. Sebaceous glands secrete sebum. Again, it's a chemical mediator. It is also a lubricant. It lubricates and protects skin from invading microbes. It is a food source for your resident microbiota that live on your skin. So, yes, some of them eat the sebum. And then the resident microbes produce oleic acid, which is an exogenously produced chemical mediator. Antimicrobial peptides, AMPs. These are sometimes called defensins, and they act as a variety of potential pathogens being triggered by antimicrobial peptides, AMPs, sometimes called defensins. These act against a variety of potential pathogens being triggered by sugar and protein molecules on the external surfaces of microbes. Researchers have figured out several ways in which antimicrobial peptides work. Some punch holes in the cytoplasmic membranes of the pathogens. Others interrupt internal signaling or enzymatic reactions. Some AMPs are chemotactic factors that recruit leukocytes to the site of infection, and others assemble to form fibers and microscopic nets that trap the bacteria. The complement system is a set of serum proteins designated numerically according to the order of their discovery. These proteins initially act as opsonins and chemotactic factors and indirectly trigger inflammation and fever. The end result of a full complement activation is lysis of the foreign or pathogenic cells. Complement is activated via the classical pathway. This is where the antibodies activate the complement. The alternative pathway, this is where the pathogens or pathogenic products such as bacterial endotoxins and glycoproteins activate the complement. And then in the lectin pathway, microbial polysaccharides bind to acute phase proteins. This is what it looks like. The classical pathway begins with an antibody opsonizing. And by the way, when you hear opsonize, opsonization, that just means that it's coating something, it's covering it, okay? So the classical pathway begins when the antibody opsonizes or coats the microbe, and this is followed by recruitment and activation of the C1 complex here. So the C1 is a serum protein, and it's called C1 because it was the first one that was discovered. Then the rest of the cascade becomes activated. So you have here is the lectin, the beginning of the lectin pathway. This is when a mannose binding lectin opsonizes the microbe, and then from there the pathway is virtually identical to the other classic cascades. It's basically just the alternative pathway is spontaneously activated, and then the C3 complement protein react to the presence of the invader, and then the C3 complement will latch onto the invader and it coats it, it opsonizes it, and begins the activation of the rest of the cascade. So from the beginning, the 3 complement activation pathways, they have different triggers, but they all result in the activation of the complement protein C3. C3 produces C3A and C3B. C3B binds to the surface of the target cell, the pathogen cell, and it works with the other complement proteins to cleave C5 into C5A and C5B. And then C5B binds to the cell surface and recruits C6 through C9. And C6 through C9 form a ring structure called the membrane attack complex, the MAC. The MAC punches a hole through the cell membrane of the invading pathogen, and that causes the cell to swell up and burst like a bubble. Here is a quick video to help illustrate this phenomenon.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5A and C5B. C5A enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Cytokines are proteins that facilitate various non-specific responses by innate immune cells. So this is going to be your production of other chemical mediators, your cell proliferation, cell death, and differentiation. Acute phase proteins change their serum concentration in response to inflammation. Histamines respond to injury or allergies or inflammation. They cause contraction of smooth muscles and dilation of capillaries. Your leukotrienes are isolated from leukocytes. Prostaglandins cause contraction of smooth muscle. They dilate blood vessels. They control blood pressure and modulate inflammation. And then bradykinin also cause contraction of smooth muscle, and they dilate blood vessels. So these are all different kinds of cytokines. Cytokines play a very key role in inflammation, and they trigger production of inflammation eliciting mediators. This here has 3 different classifications of cytokines. So your autocrine, these are cells that both secrete and receive the signal. Paracrine, they secrete the signal and then it's received by a nearby cell. In endocrine, they secrete the signal and then it goes into the bloodstream and travels to cells that are far away. Blood is a complex liquid tissue composed of cells and portions of cells within a fluid called plasma. Plasma is mostly water. It contains electrolytes, dissolved gases, nutrients, and lots of proteins. Cells and cell fragments suspended in the plasma are called formed elements. In a process called hematopoiesis, blood stem cells located principally in the bone marrow within the hollow cavities of the large bone. They produce 3 types of formed elements. These are the erythrocytes, platelets, and leukocytes. Erythrocytes are the most numerous of the formed elements. They carry oxygen and carbon dioxide in the blood. Platelets are pieces of large cells that are called megakaryocytes, and they have been split into smaller portions of cytoplasm surrounded by cytoplasmic membranes, and erythrocytes are carrying the oxygen, okay? Platelets are the things that are involved in blood clotting. And then leukocytes, these are another type of formed element. These are basically what we're talking about when we're talking about white blood cells. They call them white blood cells because when you separate blood in a test tube, the leukocytes kind of have this white color. There'll be like a whitish layer in the test tube. Based on their appearance and stained blood smears, when viewed under a microscope, leukocytes are divided into 2 different groups. So you have granulocytes and agranulocytes. Granulocytes are leukocytes that have lobed nuclei and granules. And then agranulocytes are mononuclear and they do not have granules. So figure 1714 is showing some micrographs of your granulocytes. The first one here with several lobes is a neutrophil. Then in the middle here, it has 2 lobes. That's an eosinophil. And then the last one that doesn't have lobes is a basophil. Back up for a second here and talk about lymphocytes. Lymphocytes are the T cells and the B cells. So these are going to be really important, especially in the next lesson when we're talking about adaptive immunity. And then you have your monocytes. These are the phagocytosis cells, and they also are your antigen-presenting cells. And of course, we talked about platelets. This has to do with clotting. And it also has to do with homeostasis and growth factors. Mast cells are similar to basophils. They induce and promote inflammation. This figure shows mast cells in blood, and in the blood smear, they are difficult to differentiate from basophils. But unlike basophils, mast cells migrate from the blood and move into various other tissues. So that is one very distinct feature of mast cells. Natural killer cells are called NK cells. These are inhibited by the presence of MHC. MHC stands for major histocompatibility cell. MHC receptors are normally on healthy cells, but if you have cells that are infected or otherwise abnormal, so things like cancer cells, they have a reduced expression of the major histocompatibility cell receptors, the MHCs. they have fewer of them, and they have more activating molecules. So what happens is, in a healthy cell, so here you have your NK cell, healthy cell. The healthy cell has plenty of MHC, and the NK cell has an activating receptor and it has an inhibiting receptor. The inhibiting receptor attaches to MHC. If it's there, then everything's fine and the healthy cell stays healthy. But in an unhealthy cell, the MHC is not there, but a lot more of the activating receptors are there. And so what happens is the NK cell, with its lack of MHC to bind to, will then bind here, and then that activates the NK, the natural killer cell, to kill that unhealthy cell. Monocytes are large, agranular white blood cells with a nucleus that lacks lobes. Okay, so we already know that. When the monocytes leave the bloodstream, they differentiate and become macrophages. Macrophages are large white blood cells, and they are a type of phagocyte, and they lurk particularly in places that are vulnerable to invading organisms, so places like the lungs and the spleen. Macrophages possess cell surface receptors that can identify and stick to some molecules on the surface of pathogenic bacterial cells. So your macrophages are scavengers. They recognize and engulf and digest pathogens and other microbes, especially those that live inside of the host cells. Phagocytes affix themselves to microbes and cover them with proteins. Phagocytes consume the microbes and then they chop them up with enzymes. And the phagocytes plaster their cell surfaces with pieces of the microbe that they chopped up, and these are known as antigen-presenting cells. The antigens are attached to the major histocompatibility cell complex. So the antigens come from invading microbes that got chopped up by phagocytes. And the phagocytes presented these antigens all around the outside of their own cell membrane. And then those antigens become attached to MHC proteins. And it's like putting them on exhibit. And this permits them to be recognized as antigens by other cells that are in the immune system. Your toll-like receptors, the TLRs, these act as early warning systems. They trigger your body's responses to a number of molecules that are shared by various bacterial or viral pathogens, but they're not normally found in humans. So the microbial molecules that we're talking about here are going to be your peptidoglycan, the stuff that's prominent in gram-positive bacteria, your lipopolysaccharides, which are in gram-negative bacteria, also things like flagellin, unmethylated pairs of cytosine and guanine nucleotides that came from lysed bacteria in virus cells, and other weird stuff like double-stranded RNA or even single-stranded RNA, but it came from viruses. And then all of this is collectively referred to as the pathogen-associated molecular patterns. So PAMPs, pathogen-associated molecular patterns. Okay. So this is an interesting figure. I'm going to see if I can kind of help you understand what's happening here. You have damaged cells. You have macrophages. The macrophages invested pathogens, and that released cytokines that are causing inflammation, and it also released cytokines that are chemotactic for leukocytes. So there's a lot going on here. You have activation of a complement, then you have production of chemotactic and inflammatory C5a, and then you have leukocytes that are leaving the blood vessel, and they follow the chemo-attracted signal to the cytokines in the C5a to the site of infection. And then you have granulocytes, like neutrophils, that are releasing more chemicals that destroy the pathogens. And then you also have phagocytosis and intracellular killing by NK cells. So there's a lot going on here. But essentially, it is showing how different parts of your innate immune system work together to recognize, track down, engulf, or trap, and then digest or kill invading pathogens. Phagocytosis is what they are talking about when they say that pathogen cells are getting digested. Phagocytosis actually means eating by a cell. They're eating the invaders. Phagocytosis is a complex process. I know, big surprise. But it is a complex process, but it's basically, it's still not completely understood, but it's basically 6 steps. You have step 1, chemotaxis. Step 2, adherence. Step 3, ingestion. Step 4, maturation. Step 5, killing. And then step 6, elimination. Chemotaxis is the movement of a cell either toward a chemical stimulus. So if it's moving toward a chemical stimulus, this is positive chemotaxis. Or movement of a cell away from a chemical stimulus is called negative chemotaxis. In the case of phagocytes, positive chemotaxis involves the use of pseudopods to crawl toward the microorganisms at the site of infection. Adherence includes the engulfment or opsonization of a pathogen with antimicrobial proteins, such as the complement proteins that we've seen, the C1 through C9, and or antibodies. So the coating process, that's what we're talking about when we call it opsonization. It's coating, it's engulfing, it's like wrapping around. That's opsonization and adherence. The encompassed microbe is internalized by fused pseudopods, which form a food vesicle called a phagosome. So that's here. The phagosome is here. This is the invading bacteria. This is the phagosome. This whole thing is the macrophage. A series of membranous organelles within the phagocyte fuse with newly formed phagosomes to form digestive vesicles. So that's what is happening here. One organelle, called the lysosome, adds digestive chemicals to the maturing phagosome, which is now called a phagolysosome. So that is a phagosome that has merged with a lysosome. It's called a phagolysosome. Phagolysosomes contain antimicrobial substances such as highly reactive, toxic forms of oxygen, lipases, proteases, nucleases, and all of these enzymes destroy the engulfed microbes. Digestion is not always complete, and phagocytes eliminate the remnants of microorganisms via exocytosis. Exocytosis is basically the reverse of digestion. Phagocytic cells contain pattern recognition receptors, PRRs. PRRs recognize various PAMPs. Remember, PAMPs are pathogen-associated molecular patterns. So what this is saying is that the cells that conduct phagocytosis, they have these receptors that recognize the patterns that are found on pathogens. And when the PRR recognizes a PAMP, it sends a signal to the nucleus that activates phagocytosis and cell proliferation and production and secretion of antiviral interferons, pro-inflammatory cytokines, and the NK, the natural killer cells. Inflammation is a general non-specific response to tissue damage resulting from a variety of things like heat, chemicals, exposure to UV light, abrasions, cuts, and of course pathogens. You have acute inflammation, chronic inflammation, and fever. Acute inflammation develops quickly, it's short-lived, it's typically beneficial because it results in the elimination or resolution of whatever condition caused it. Acute inflammation results in dilation of and increased permeability of blood vessels and migration of phagocytes and tissue repair. Long-lasting chronic inflammation causes damage and sometimes death to tissues and can result in disease. Both acute and chronic inflammation exhibit similar signs and symptoms, including redness, localized heat, edema, and pain. Fever is a body temperature above 37 degrees Celsius, or 98.1 degrees Fahrenheit. Fever augments the beneficial effects of inflammation, but like inflammation, it also has unpleasant side effects, including malaise, body aches, and tiredness. The hypothalamus, which is a portion of the brain just above the brainstem, it controls the body's internal core temperature. Fever results from the presence of chemicals called pyrogens. Pyrogens are bacterial toxins or cytoplasmic components released due to lysis, cell lysis. Fever results from the presence of the pyrogens trigger the hypothalamic thermostat, and it then resets at a higher temperature. Part of the body's initial response to an injury or invasion of pathogens is localized dilation of blood vessels in the affected region. The process of blood clotting triggers the conversion of a soluble plasma protein into an amino acid peptide called bradykinin, which mediates inflammation. Basophils, platelets, and mast cells also release inflammatory mediators such as histamine when they're exposed to complement fragments. So the figure here is showing that histamine increases blood flow to the wound site, it increases vascular permeability, and this allows for fluids, proteins, phagocytes, and other immune cells to enter the infected area. But this results in swelling and redness of the site, and it increases blood flow to the injured site and causes it to feel warm to the touch. Inflammation is also associated with pain because all of these events, all of these things that are happening, stimulate nerve pain receptors in the tissue. A tubercle is a granuloma in the lung tissue of a patient with tuberculosis. So this here is a granuloma, a tubercle. A granuloma is the term for a small area of inflammation. A tubercle is a granuloma in lung tissue. And this is lung tissue taken from a patient that has tuberculosis. And the purple things here, these are the white blood cells that have surrounded and walled off this granuloma. It is important to note that even though in this case they're calling this a tubercle and it's from a tuberculosis patient, granulomas occur in many other forms of disease besides just tuberculosis. So that's going to do it for lesson 1. I want to thank you guys for watching. Don't forget to do the reading. Check the description below for more videos related to these topics, and leave your questions for me in the comments section below.